Hi, everybody. I hope you've had a fantastic festive season. Happy New Year. Welcome to another Chats with Children into 2021. And I'm delighted today to be joined by Jeffrey Silva, who is the Director of Product Development from Adeptrix Corporation. And today we're going to be talking about BAMS, which stands for Bead Assisted Mass Spectrometry. But before we do that, let's introduce our guest. Jeffrey, Happy New Year. How are you, my friend?
Very good. Thank you so much. Happy New Year to you. I appreciate the opportunity for this interview and to share with everyone news about BAMS and what's happened with Adeptrix over the past couple of years.
Fantastic. Well, look, before we start talking about BAMS and what it means and what it is, perhaps you tell people a little bit about Adeptrix, who are not familiar with the company, and also your role within that.
Sure. Adeptrix has been around since about 2010. It really didn't focus on the key app until about 2015, which is targeted proteomics. I got involved with it about that time because of my history. I'll sort of get into that in a little bit. But Adeptrix is focused on creating tools for easy to use targeted proteomic applications, coupling immuno affinity capture with MALDI mass spectrometry for sort of a high definition targeted proteomics applications. In terms of my background, I was at Waters for in early 2001-ish, spending most of my time doing electrospray, proteomics, working on discovery-based protein analysis, and on technology that is now being used for a lot of discovery-based proteomics called SWATH. At the time, it was data independent analysis. And then from there, because of my interest in targeted proteomics and post-translational modifications, I went and joined a company called Cell Signaling Technology they were out of Danvers, to really get a better appreciation of post-translational modifications and analyzing them with mass spectrometry, coupling antibodies for that type of work. And I was there for about 8 years, leading a group that did service work using those reagents to carry out those types of studies for pharma, biotech, and academia.
Brilliant. So what is BAMS? Tell me what it is.
So BAMS, as you said earlier, stands for Bead Assisted Mass Spectrometry, and what we've done is we've created a technology that where we coat individual beads with antibodies, and these beads act as molecular tweezers to capture the target analytes of interest, so that and enrich them on the bead itself. That can then be used to deposit the material onto a solid surface for mass spec or MALDI analysis. And by doing so, what we've done is we've eliminated the complicated LC front end of the mass spec and now LCMS workflow, and are using affinity capture with MALDI MS for targeted proteomics.
Right. Okay. And what are then the challenges around targeted protein analysis, and what methods have researchers historically used to address them?
So the challenges are that, at least what we've seen and what people can easily find with historic data and public proteomic data, is that proteins exist in multiple forms. Any given protein, in the majority of cases, don't exist in a single form. And what I mean by that is, as we all know, protein is a linear sequence of amino acids, right? But that protein doesn't exist as just a linear portion, a sequence of amino acids. There can be truncated forms of that protein. There can be post-translational modifications like phosphorylation, acetylation, methylation. These act as switches and dials that really modulate the activity, the function of proteins, where they're localized, what the activity is, whether it's inactive or active, whether it's in the nucleus or in the cytosol. And so a given protein can exist in multiple forms. And so what BAMS allows for is us to target specific locations of the protein and look at the variants within that region and be able to monitor those different proteoforms in response to different perturbations, whether it be comparing normal and diseased tissues, or whether it being looking at the response of a cell with a particular drug and how it reacts to how the protein or the modulation of that protein responds to the presence of that drug. In terms of what technologies are used today, typically people are using ELISA-based methods, which are very popular assays. They're easy to use. The challenge there is that they're typically a low resolution assay. And what BAMS does is it couples affinity capture with a high resolution detection so that you can see all these different features within the context of a particular protein.
Right, brilliant. And what other applications then can you apply BAMS to?
So we've with COVID this year, we had previous to this, we had been building up reagents and assays to areas like oncology, looking at specific phosphorylation on specific proteins, acetylation, methylation. We've been looking at neurology, looking at degradation products in beta amyloid, looking at proteins like tau that's associated with Alzheimer's disease, and how tau and hyperphosphorylated tau is associated with that disease, and designing assays to be able to monitor those different features. Earlier this year with COVID, we decided that we could adapt the method to be able to look at viruses at COVID as a way to detect the virus. And this was simply done by, because the reagents were available, the affinity reagents were available, we took affinity reagents to spike protein and coupled them to the bead as a way to capture the spike protein. And the reason why we focused initially on the spike protein was that the spike protein is on the surface of that virus. So it provides a mechanism to be able to capture the intact virus and be able to place that material on the surface for MALDI analysis. Right. There's an instrument or a platform out there called that Brooker has created called the Biotyper, which is a tool for microbiology for identifying microbes. And by the way they do that is they grow colonies and then transfer the colony by hand with a toothpick or a sterile applicator, and transfer it onto the surface of the MALDI plate and literally shoot it with the laser and obtain a fingerprint for that microbe and compare that against a library to identify the microbe. The challenge with viruses is you can't grow them up. You can't grow them up as a colony that you can then physically transfer over to the plate. And so what BAMS does is it acts as that device that's able to capture, concentrate, and deliver the material onto the surface of the plate, whether it be the protein component itself in the liquid biopsy, or the intact virus itself onto the surface of the plate for analysis. And so that's been something that we've been working on. We've expanded it beyond the spike protein to include nucleocapsid. And because BAMS is able to do multiplexing meaning we can target many different proteins just by using different beads with different affinity reagents, we can carry out an assay that can, a platform that can be used for virology to look at many different types of viruses.
Right. Well, that leads on nicely, actually. So where do you see the future of BAMS then?
The future of BAMS, in terms of what our vision, in terms of what we hope BAMS to do, it's really to put proteomics in the hands of many more scientists, biologists, and not to be feel the need that you have to be an expert in mass spectrometry, because what we've done is we've built the technology on something that's very familiar, magnetic bead capture with MALDI-MS, which in essence is a scanner. In this format serves as a scanner, because when we carry out and characterize the reagents, Adeptrix identifies and creates the reagent to say that this bead enriches for this protein and generates this mass. And so that can be coded in software so that people don't have to look under the hood and look at mass spec data. The software can simply tell you what it is you've detected and do the quantitation as well. And so the goal really is to create an easy to use targeted application to enable targeted proteomics for biologists, and in terms of the future, really to expand it in a number of different areas and have that driven by customer use and customer excitement about the technology. So oncology is an area that we see a lot of growth potential. Neurology, we've got a number of different collaborators that are in the neurology space that see the value of being able to monitor these different proteoforms and also expand the content to include other protein targets that are needed to stratify different neurological disorders. And so we'll be building up content on that. Epigenetics is another area that's really exciting, because histones, the tails of those histones are dressed with a lot of these post-translational modifications, these switches and dials, right? And we can capture those pieces and look at all those different features and correlate that to developmental biology, disease biology, drug treatments. And so that's something that we're really excited about, because the reagents that are out there that we're working with are very good, and it really exposes a lot of interesting characteristics about those the histones.
Brilliant. So finally, because I've been asking everybody this question, if you're looking back at 2020, what's one word you'd use to describe the year that's just gone? And looking ahead, how do you see 2021 developing?
I guess the word that comes to mind is perseverance, really. You know, it's been a tough year. It was a tough year. But, you know, with that, we really just focused on what we needed to do to refine the application, get the message out there through our relationships with our collaborators, participate in these virtual conferences. That was not quite the same, but we still, you know, pushed through and communicated that information. We published on the work as well. And so, you know, we're happy with the progress that we made. And with regard to 2021, because of that perseverance, that hard work, that focus, it really has led to an exciting announcement that will happen soon. And unfortunately, I can't say it at the time because it's not released, but we do have a major investor partner who's been sort of with us for a while watching the development of the technology, and we're excited to have them on board with us to help introduce this technology to more people and help us spread the news about BAMS.
Brilliant. Well, and I'll say to viewers that as of this recording, that announcement has been made, hence the reason why it's not coming on the video. But if the announcement is or has been made by the time the video goes live, then the link will be below the video, or above the video, I should say, in the actual post. You'll be able to click on that and see what that announcement was. So a little teaser in the video that you can then click on the link from that shows how live this recording is. So there you go. and also, if you want to know more about BAMS, where can they get more information and find out more?
We've done, you can find more about BAMS by going to the Adeptrix website, www.adeptrix.com, and there's a resources tab on the company website that has announcements, posters, publications, tech notes, application notes, and there's also a contact us page that you can fill out, and we're happy to follow up with a conversation and provide you with more information about the technology.
Great. And as always, to save you guys all having to write everything down, the links, as Jeffrey's just described, will be again above the video to save you having to do all of that hard work. So I'll put those links in for you to save you doing the effort. And I'm sure Jeffrey won't mind if you've got any questions based on what Jeffrey's talked about today, then feel free to put them in the comments section as well below the video, because it's always great to hear what people have thoughts about this technology and the interview that we've done. And I'm sure Jeffrey will be happy to answer them, or you can, I'm sure, message him directly on LinkedIn as well.
Absolutely.
So Jeffrey, what I say is thank you very much for taking time out to talk to me today. It's a really interesting subject, great technology. So I wish you and the company lots and lots of success and a wonderful 2021. So, and also obviously your family and your loved ones.
Thank you so much. Really appreciate it.
No, I really do. Thank you very much for taking time to talk to me. As always, everybody, thank you for watching. If you've got any comments, please comment below the video, because it's great to know what people think about these videos. So please get involved and engage. It'd be great to know what your thoughts are. And as always, as it is the start of the year, I wish you and your loved ones a fantastic 2021, a happy, healthy and successful 2021. And as always, until next time, stay well, stay positive, stay safe. Thanks a lot. Bye-bye.